Welcome to AK Bio lecture series.
The DNA is an amazing molecule in many ways, and if stretched out in a thin thread, it would measure around 1.8 meter in length. Each and every cell is less than a tenth of millimeter in size only. The DNA must be replicated and segregated precisely in order for cell to divide and create two daughter cells. Key to this process is the chromosome. So, proper compaction of the DNA into chromosome is essential part of the cell cycle. In the last class, we discussed the stages of the cell cycle in general. They are interphase and M phase. Interphase is again divided into G1, S, G2 phases. We also discussed about the changes that happening to the chromosome in terms of number of chromatids present before and after the S phase, that is the replicating phase. If you have any doubts, then you can refer this chart also. In this chart, the number of chromatids, chromosome and DNA at each phase of the cell cycle is well explained. So, after the S phase, the DNA is replicated. Then the daughter double-stranded DNA and the parental double-stranded DNA become attached at the centromere region. But remember one thing, they are not at all compacted. They are thread-like in structure. So, but remember one thing, they are not at all compacted at the end of S phase and even in G2 phase. How the DNA is compacting or packed into the nucleus during the progression of the cell cycle, and which are the minute changes that occurs during the progression of cell cycle will be discussed in this class. In this class, we are going to emphasize on the mechanism of DNA compaction in prophase of M phase. So let's see the events in prophase. How many number of DNA you can see as part of a single chromosome when the cell just enters the M phase? How many number of DNA you can see as part of a single chromosome when the cell just enters the M phase? I hope you know the answer is 2. Yes, this is the one DNA and this is the another DNA. I hope you know the answer is 2, and if any doubt, please go back to the first slide of this class or the previous class with the table explaining the same. As the prophase progresses, the duplicated DNA undergo condensation. The chromatin or the DNA undergo condensation process by tight coiling of each double helix. So, each duplicated chromosome appears as two identical sister chromatids. So then several changes will occur to the cell when it passes through the prophase. We can see the chromosomes like structure, but it's not exact chromosomes, chromosome like structure, but not the fully formed or compacted chromosome at this stage of cell cycle. Means chromatin condenses when the phase progresses. This is the first major change of this phase. Actually, the process of compaction of DNA is a wonderful process The details will be discussed in coming slides. The second event of this stage is the disappearance of nucleoli. So this is the first event and this is the second event. Okay. The third event is the disappearance of nuclear membrane. Nuclear membrane becomes small vesicles then joined with ER membrane. By this way, dissolution of nuclear membrane takes place. In this slide, you can see here the changes in each stages of the cell cycle. Here in interphase, you can see the nucleus here and ER here. ER is located very near to the nucleus with some attachment with the nucleus. Here and there, you can see some attachment with the nucleus. As the cell cycle progresses into the prophase and prometaphase, or in between prophase and prometaphase, you can see the late prophase. You can see the fusion of the vesicularized nuclear membrane with the ER membrane as seen here. In pre-metaphase or late prophase stages. I am repeating the point once again. During prophase, the nuclear pore complex disassembles and the nuclear envelope separates from the nucleus and merges with the endoplasmic reticulum. This organelle is then partitioned and inherited by daughter cells. This is how nuclear membrane disappears. It is a very crucial step in the process of compaction of DNA into chromosome. We will discuss the relationship between the disappearance of nuclear membrane and DNA compaction in coming slides. Actually, we discussed 3 events in prophase. The fourth event is kinetochore maturation. In this slide, in the diagram, you can see the chromosome as such here, and a particular portion of centromere of this chromosome is enlarged into this much details. You can see the centromere and kinetochore here. Okay. Then what is kinetochore? Kinetochore is a complex of proteins associated with the centromere of chromosome, to which the microtubules, you can see here the microtubules of the spindle fibers attached during cell division. And kinetochore is a complex of proteins associated with centromere of a chromosome, to which the microtubules of the spindle fibers attached during cell division. As part of kinetochore maturation, kinetochore assembled at the centromere region. Without centromeres, cells cannot divide properly, and the overall process of mitosis and meiosis fails. The centromere is a region of specialized chromatin, that is constitutive heterochromatin, that provides the foundation for kinetochore assembly and as a site for sister chromatid attachment. The kinetochore itself have three sets of subcomponents. Three sets of subcomponents. In the picture B, you can see the three subcompartments and it is labeled as A, B and C. This first subcompartment is A, which is named as the chromosomal DNA inner kinetochore protein interface. Once again, I will tell the chromosomal DNA inner kinetochore protein interface, where from the name itself you will get the idea where the DNA is the region where the DNA of the centromere is attaching with the kinetochore, and the second compartment is B. The name is the inner kinetochore mitotic spindle interface. Inner kinetochore mitotic spindle interface. From the name itself, you will get the idea or outline where the kinetochore and spindle fibers will get attached in metaphase stage. And third one is represented by C. The name is the kinetochore protein cell cycle machinery interface, where the cell cycle regulatory proteins such as checkpoint proteins, anaphase promoting complexes interacts with the kinetochore. So the third one is kinetochore protein cell cycle machinery interface. From the name itself, you will get the overall picture where the cell cycle, it is the place, it is the portion, it is the subcomponent of the kinetochore where the cell cycle regulatory proteins such as checkpoint proteins and anaphase promoting complexes interact with the kinetochore. These are the three subcompartments of kinetochore. The assembly of these proteins is the fourth event in prophase of cell cycle, that is called maturation of kinetochore. The fifth event in prophase is the formation of mitotic spindle. In the picture, the green colored portion is the mitotic spindle, which is radiated out from the central core. I am changing the color of the ink. You can see here the central core. From the central core, the green colored portion, that is the mitotic spindle, is radiated out. The central core is called centrosome. The radial array of shorter microtubules that extends from the centrosome, this is the centrosome, and these are the radial array of shorter microtubules, and these are called asters. The centrosome move away from each other when the cell cycle progresses by oppositely acting pressure as part of lengthening of microtubules. And these are the five major events in prophase. Then, as the first event in the prophase, we discussed the process of compaction or condensation of DNA into chromatin and chromatin into loosely packed chromosome. I am using the term loosely packed chromosome because it is not a matured compacted chromosome in prophase. Only in metaphase you can see the compacted, highly matured chromosome. In the case of prophase, it's the early stages of it is in the early stage of compaction process. So you can see only the loosely packed chromosome. But in the very initial stage, it is not that much loosely packed chromosome. It is very highly relaxed thread-like structure. So different proteins are involved in this process of compaction of DNA into the chromatin and then into the loosely packed chromosome. So we will see those proteins in coming slides. There are two kinds of proteins actually which are acting on the DNA in the initial stages of the compaction. They are cohesin and condensin. And this is the structure of cohesin and this is the structure of condensin. They belong to SMC group of proteins. What is SMC? SMC stands for structure maintenance of chromosome. So they belong to SMC group of proteins, and these proteins cohesin and condensin helps in the maintenance of the structure of chromosome. They are, from the picture itself, you can understand that this is a multi-subunit protein. These are a multi-subunit protein, and cohesin is made up of 4 subunits, that is SMC1, SMC3, SCC1, SCC3. And while condensin is made up of 2 subunits, that is SMC2 and SMC4. These are major subunits of condensin. You can see here another proteins which are functioning as linker or linker domain. For easy remembering of the number, cohesin is composed of proteins with odd number 1 and 3, and condensin is with even number 2 and 4. So when we look at their function of cohesin and condensin, both are involved in compaction of the DNA, but the mechanism of action and location of action is little bit different. Suppose when a cell has completed the S phase, now one DNA, okay, 1 minute. When a cell has completed the S phase, now one DNA is duplicated into 2 DNA, that is 2 sister chromatids. Can represent it as this one also. So single-stranded chromosome is now double-stranded chromosome, now replicated into double-stranded chromosome, which are connected at the centromere portion. So these 2 chromatids are called sister chromatids. Okay. So 2 sister chromatid strands formed with some attachment between them. Usually what happens is that the DNA at this point is not that much coiled, they are well relaxed. So the DNA is very relaxed. It's not at all compacted one. So They are lying side by side as I draw, and they are lying side by side, but they will move apart or away freely in any direction as a simple thread when flying on the air, just like that, if they are not bounded with each other properly. So they are lying side by side, but they will move apart or away freely in any direction if they are not bounded each other properly. If they are not bounded means if the chromatids, sister chromatids are not bounded each other properly. If it moves as free thread, what will happen? They will coil in wrong way, and also the chance for the breakage of the DNA due to tension created by movement and torsions. So there is a chance for aberrations and loss of genetic material from the system. So proper coiling is essential for the chromosome segregation in dividing cells. So the newly formed double-stranded DNA and the parental double-stranded DNA, here you can see the cell. Consider this is a cell, this is a chromosome after S phase. Okay, so you can see here the parental DNA and daughter DNA. Both are double-stranded, and so the newly formed double-stranded DNA, that is daughter DNA and parental double-stranded DNA, must be connected together. Here comes the role of cohesin. Okay, this protein hold the newly replicated and parental DNA together. Means cohesin holds the newly replicated sister chromatids together. Okay, that is the function of cohesin. Cohesin's function, what is the function of cohesin? Cohesin holds the newly replicated and parental DNA together. So they hold the sister chromatids together. So the cohesin protein is located throughout the length of the chromatid, and they are connected with each other. Okay. Before entering into the details of cohesin cycle, you must keep in mind that for easy way of communication and understanding, we are talking about one chromosome out of 46 chromosomes in a single cell in the case of human. Keep in mind that all the things we are discussing about the single chromosome is applicable for 46 chromosomes, and all the events we are discussing in simultaneous is simultaneously happening to the 46 chromosomes in a single human cell during cell cycle, even though we are discussing about a single chromosome. The mechanism of cohesin action is very interesting to understand. In this slide, you can see G1, S, G2 and M stage of the cell cycle, and chromosome is represented in blue color. You should note that the number of chromosomes and chromatids vary according to the stage of the cell cycle. You must have the idea of chromatids and chromosomes in each stages of the number of chromatids and chromosomes in each stages of the cell cycle. And if you don't know or forget the details, then go back to the first part of the video or the previous lecture and then learn it thoroughly and come back. I am not repeating it here. In this picture, the length and compaction of the chromatids. Here you can see the chromatids. Here also you can see the chromatids. So in this picture, the length and compaction of the chromatid and chromosome is not scientifically represented. Actually, the DNA at S phase and G2 phase is highly relaxed and fine thread-like. Okay, but when the cell enter M phase, when cell enter M phase, that is in the beginning of the prophase, DNA began to condense into chromatin and then condensed into semi-chromosome like structure. Okay, then first in G1 phase, one chromosome with one chromatid is present. In the S and G2 phase, one chromosome with two chromatids is present, and then in M phase, two chromosomes and two chromatids can see depending on the stage in M phase. Up to anaphase, the chromosome is with two sister chromatids which are attached at centromere. But in anaphase, the spindle fibers split the sister chromatids from the chromosome and moved to opposite poles of the cell. So at this stage, you can see the 4N condition within a cell during the mitosis. Okay, the process of cohesion, cohesin action or cohesion can be summarized into the process of cohesion or cohesin's action is can be summarized into 5 steps. Okay, let me erase all the inks. Okay, the process of cohesin cycle can be summarized into 5 steps. The loading of cohesin protein. First one is loading of cohesin protein, and second one is establishment of cohesion, not cohesin. Cohesin is the protein, cohesion is the process, okay, or the effect of that protein. So loading of the cohesin protein. The second one is establishment of the cohesion between chromatids. Cohesion between chromatids. Okay, by cohesin. Then third is maintenance of cohesion. Then fourth is cohesin dissolution. Dissolution. Then fifth stage is completion of cohesin removal. Completion. Okay, so 5 stages. We can summarize the total cohesin cycle into 5 stages. First one, cohesin loading occurs in G1 phase in yeast or at the end of telophase of the previous cell cycle in mammalian cell. Cohesin loading is mediated by the protein NIPBL, and the second phase is establishment of cohesion. So cohesion is established between the sister chromatids by ESCO1 and ESCO2. ESCO1 and 2 proteins. It is mediated by acetylation during S phase. We are not entering into the depth of acetylation. Is the process of just know the fact that is the process of acetylation that that takes place during this stage. Okay. Then third stage is cohesion is maintained. cohesion is maintained in G2 phase. Okay, by in G2 phase. Here it is G2 phase. Okay, G2 phase by PDS5A, PDS5A and PDS5V. So cohesion is maintained throughout the S and G2 phase. S phase means the late S phase and through a full G2 phase, complete G2 phase. Okay. After the G2 phase of the cell cycle, cell reach the M phase, and at the time the chromatin condensation begins. In the M phase, we studied that as the first point, first event in the M prophase, as the first event in the prophase, we studied that condensation of chromatin takes place in prophase. So after the G2 phase of the cell cycle, the cell reaches the M phase, and at the same time the chromatin condensation begins at this stage. The process of condensation will occur independently on two chromatids. Even though they are connected each other by cohesin. Here you can see the connection between the chromatids, sister chromatids is very clear. I think for easy representation it is given as 4 cohesin that connected, which are connecting the two sister chromatids together, which are joining two sister chromatids together. Okay. Look at this portion also, because one cohesin is present at the centromere. Actually, there is no limitation or there is no fixed number. Here it is based on this picture. I am telling that only one cohesin. Actually, numerous cohesin molecules are togetherly acting on the centromere region and other parts of the chromosome. Since each chromatid coils separately, okay, like this. Since each chromatid coils separately and independently, the cohesin, here this is cohesin. Okay, since each chromatid coils separately and independently, the cohesin interconnections between the chromatids will hinder the proper coiling of each chromatids. So the green color one is the cohesin, and the green color structure, that is cohesin, will hinder the coiling of the red colored sister chromatids. Okay, I hope you can understand from the picture. So for proper compaction or condensation, the chromatids, condensation of the chromatids must be free from cohesin. So we have to remove the cohesin. I used the eraser for removing the cohesin, but in the case of cell, there is no eraser. So at the beginning of the prophase itself, cohesin should be removed from the chromatids. The process of stepwise removal of cohesin from the chromatids is called cohesin dissolution. So the stepwise removal of cohesin from the chromatids is called cohesin dissolution. Okay. Actually, the eraser in the cell, which eraser? the cohesin eraser of the cell are polo-like kinase 1, aurora B kinase 1 and VA-PL. Which are they? Polo-like kinases, aurora B kinases and WAPL protein. These three proteins, majorly these three proteins, play a central role in the cohesin dissolution process. As name indicates, kinase activity of this protein inactivates the cohesin by the phosphorylation of this protein. So, polo-like kinases, aurora B kinases and WAPL proteins phosphorylate the activated form of cohesin, and by the phosphorylation process they will get inactivated. Okay, the phosphorylation will inactivate the cohesin protein. As a result, it will fall off except at the centromere region, where most of the cohesin protein remain protected at the centromeric region by SGO1 protein, by the phosphatase activity of SGO1. So the SGO1 has the phosphatase activity, not kinase activity, is phosphatase activity. So SGO1 protein is present at the where centromeric region. So SGO1 protein gives the protection for the cohesin protein from the polo-like kinases and aurora B kinases and WAPL, and so these proteins, polo-like kinases, aurora B kinase and WAPL, cannot phosphorylate the cohesin protein at the centromere region. Even though they phosphorylate at the cohesin protein at the centromere region, SGO1 will remove that phosphate immediately from the cohesin molecule. So this will keep in active form, not inactive, active form. Cohesin will be active form at the centromeric region with the help of SGO1 protein, which has the phosphatase due to the phosphatase activity of the SGO1 protein. So cohesin will hold the sister chromatids at the centromere region till anaphase. So here it is prophase, this is prophase, this is metaphase, this is anaphase, and this is telophase, isn't it? So this will keep active, that is the cohesin will be active till it will reach anaphase, because only after anaphase, only after metaphase, the cohesin will be removed. So cohesin will hold the sister chromatids at the centromere region till anaphase, and cohesin should be removed from the centromere for the progression of the cell into anaphase from the metaphase. So we know that the in metaphase the chromosomes are aligned at the central portion, that is metaphase plate. Okay, here and spindle fibers is coming from here, and during anaphase what will happen, the sister chromatids will be separated by the action of spindle fibers, isn't it? So for the separation of sister chromatids, what should be essential? What should occur? The cohesin removal from the syndromic region should occur. Then only the cell can progress into anaphase from M phase. Then how this cell removes the cohesin molecule exactly at end of the metaphase stage. Cohesin is completely removed. Finally, cohesin is completely removed from the centromere by APC and separase proteins mediated proteolysis, and so finally cohesin is completely removed from the centromere by APC and separase proteins. So APC is an enzyme, adds a small protein tag called ubiquitin, that is UB. UB means ubiquitin. So when a target is tagged with ubiquitin, in general, when a target, any target, when a target is tagged with ubiquitin, what is the signal? It is sent to the proteasome. It will send to the proteasome, that is the recycle bin of the cell. Proteasome is the recycle bin of the cell. In general, when a target is any target, when a target is tagged with ubiquitin, it is sent to the proteasome for the destruction of the same, or the cell destroy the tagged element inside the proteasome complex. So APC here, what APC is doing? APC first adds a ubiquitin signal to a protein called securin, not to the separase. It adds the ubiquitin signal, destroying signal, to the protein securin. And securin, what will happen to the securin? Securin will be sent into the proteasome, that is recycle bin of the cell. What is the function of securin in normal case? Securin normally binds to and inactivate a protein called separase. Separase becomes when securin is sent for recycling, separase become, when securin is sent for recycling, separase become free, separase become active and can do its job. So separase is now active. So here you can see the active form of separase. Okay, and can do its job. What is its job? His job is, what is his job? Separase chops up the cohesin but that holds sister chromatids together. Separase chops up the chromatids, chops up the cohesin that holds the sister chromatids together at the centromere region. So only a separase can separate the sister chromatids in anaphase stage of the cell cycle. So separase is a very important protein, doing a crucial role in the progression of the cell cycle into the anaphase from the metaphase by separating the sister chromatids from the centromere. Next is condensin's action. It is very small portion when compared to cohesin, and cohesin works in the reverse manner. Reverse manner means some actions is different from cohesin. Not at all opposite function. Not at all meaning that it is opposite to the function of cohesin. Both are involved in the condensation process of the DNA into chromatin and chromosome. So, but the action, mechanism of action and the location of action is different from condensin, and the activation process and inactivation process is reverse in manner. That's the reason. Okay. When condensin becomes phosphorylated, it gets, when condensin get phosphorylated, it will get activated. When cohesin will get phosphorylated, what happened? The last slide we discussed. Cohesin will inactivated. Okay. Means when prophase starts, the same kinase that inactivates the cohesin, that is the same proteins that inactivates the cohesin protein activates condensin. Same protein that inactivates cohesin that activates condensin protein by phosphorylation. Which are the proteins? Polo-like kinases, same proteins. Polo-like kinases, Aurora B kinases, etc. So, polo-like kinases, Aurora B kinases, and WAPL separates the sister chromatids by removing the cohesin from the chromatids, and at the same time the same kinases activates the condensin protein to condense each chromatids independently. Okay. So the polo-like kinases and Aurora B kinases and WAPL separates the sister chromatids by removing the cohesin from various parts of the chromosome except the centromere region. But at the same time, the same proteins, polo-like Aurora and WAPL, activates the condensin to condense the sister chromatids independently to compact more, give command compact. That's all. Means condense. That's the command from polo-like kinase to condensin. And at the same time, polo-like kinase command to cohesin is detach from sister chromatids and facilitate the function of condensin. That's the command to cohesin. At the same time, favorable condition for condensin and unfavorable condition for cohesin. What is the favorable and unfavorable condition? Polo-like kinases phosphorylation activity is the unfavorable condition for cohesin and favorable condition for condensin. Okay. So means condensin promotes condensation of each chromatids in prophase by locking the coiled structure as seen in the diagram. Here the coiled structure is getting more and more compacted by the condense by the action of condensin, not by the action of cohesin, only by the action of condensin. Okay. Condensin promotes the chromosome compaction. Carefully listen here. Condensin promotes chromosome compaction by linking two distant segments. These are the distant segments. of a single chromatid. This is chromatid A, this is chromatid B, means this is both are sister chromatids, but this is chromatid A, this is chromatid B. So condensin promotes chromosome compaction by linking two distant segments of a single chromatid, in contrast to cohesin holding two sister chromatids together. We discussed one of the major event in prophase, which is disappearance of nuclear membrane during prophase. I hope everyone remember that event of prophase. And condensing protein gets activated only after the disappearance of the nuclear membrane. It's very interesting fact that this condensing protein get activated only after the disappearance of the nuclear membrane. How it is managed inside the cell? The inactivated form of condensin is located in cytoplasm. The inactivated form of, when this is the cell and this is the nucleus, and inactivated form of condensin is located inside the cytoplasm. Okay, cytoplasm, not in the nucleus. So only after nuclear membranes disintegration or nuclear membrane breakdown, it get access to the DNA, isn't it? The kinases, that is polo-like kinases and aurora B kinases, which are required for the activation of condensin. But the aurora B kinases and polo-like kinases all are located inside the nucleus. So they will not contact each other before the breakdown of nuclear membrane. So once nuclear membrane disappeared, nucleoplasm and cytoplasm will mix up. So at that time only the condensing protein can contact with the DNA and polo-like kinases, and also with the polo-like kinases. This is how the cell makes sure that there will not be a premature compaction of DNA into chromosome. That means the DNA will only get compacted, only starts to compact or condense when it reaches to prophase. That's the wonderful engineering by the cell, of the cell. When some phosphatases, the last point is, when some phosphatases acting on phosphorylated condensin, the phosphorylated condensin means active form of condensin. Active form of condensin means that promote the condensation of the sister chromatids. So when any phosphatases acting on the phosphorylated form of condensin, that means active form of condensin, what will happen? The phosphatases will remove the phosphate group from the condensin, and at that time the condensin will be inactive, and at that time what will happen? So the phosphate group will get removed from the activated form of condensin, then the compacted DNA will transform back to less condensed form. This is how compaction of DNA takes place in prophase. This is how compaction of DNA takes place in prophase.
A protein complex called cohesin holds sister chromatids together until the chromatids are separated at the start of anaphase in mitosis or meiosis. Cohesin complexes first connect the arms and centromere of sister chromatids at the end of DNA synthesis before chromosomes are fully condensed. The cohesin complexes keep the arms attached as the chromosomes condense in early prophase. Then the cohesin complexes holding the arms together release by the middle of prophase, leaving the sister chromatids connected only by the effect of cohesin complexes at the centromere. The transition from metaphase to anaphase requires the degradation of cohesin at the centromere, allowing the sister chromatids to separate.
This is another picture representing the events we discussed. You can go through this also and try to relate with the things which we discussed in last 40 to 45 minutes, and so that is the end of prophase of cell cycle. We will discuss the next phases in coming classes, and thank you.